Good morning, everyone, and welcome. Welcome to our 28th annual lead conference. My name is Nancy Redke, and I'm the field coordinator for the Montefiore Medical Center Lead Poisoning Prevention and Treatment Program, which is the sponsor for today's event. So thank you for joining us as we explore together why lead poisoning is still a lifetime challenge. We also want to thank the New York State Department of Health for making it possible to include so many of our colleagues, almost 400 now, all across this country, not just those in New York State this year. I feel as though for those of you on the West Coast, though, we should be airdropping cups of coffee at this point. A special welcome to you for getting up so early to join us. So thank you. Now, just a quick note about the morning. Obviously, your mics are muted, but please remember, you have the Q&A box in order to ask questions at any point during the presentations today. We will be monitoring it, and after each presentation, we will get to as many of the questions as possible. But we are also recording, so you will be getting the link as soon as it's available after the event today. So just keep that in mind and ask questions as they occur to you in the Q&A. Now, before we get started, I'd like to just briefly share a little bit about our program, especially for those of you outside of New York State. The photo that you saw earlier is a fitting place to start because Dr. John Rosen was clearly a pioneer in the field of lead poisoning prevention and treatment, and also the founder of our program. He began his international career right here at Montefiore in 1969, over 50 years ago. And from the start, he recognized the need for a multidisciplinary approach to lead poisoning prevention and treatment. And as a result of his vision and his hard work, the program is now distinguished by a team consisting of professionals in medicine, research, social services, environmental investigation, and public advocacy. And since 1993, we have also been a New York State designated lead resource center. A year before that, Dr. Rosen also established our first Safe House, a residential facility that now boasts 6 apartments and also houses our offices. This is a place where families whose homes are being remediated for lead can stay in a lead-safe environment while the work is being done. I should note that our program from the beginning has been a clinic-based program. And also, we are fortunate to have a laboratory that runs 20 to 30,000 lead tests a year. So with that as background, it's my great pleasure to introduce today a colleague of Dr. Rosen's and our current director, Dr. Maury Markowitz. Dr. Markowitz received his medical degree from Albert Einstein College of Medicine, and since 1978, he's been attending physician at our Children's Hospital and professor of pediatrics at the Albert Einstein College of Medicine. In 2012, he became the second director of our lead poisoning prevention and treatment program, and also the director of our lead resource center for the state. On the national level, he spent 6 years as a member of the CDC Lead Advisory Panel that developed the guidelines for the evaluation and management of lead exposure during pregnancy and lactation. And on the state level, he is also a member of the New York State Department of Health Lead Advisory Council, and locally in New York City, a member of the Healthy Homes Advisory Committee. To summarize his numerous credentials, Dr. Markowitz has had over 40 years experience as a clinician, scientist, and educator, focused primarily on lead poisoning, as well as an active researcher with over 20 years of NIH funding. Internationally recognized for his expertise, He collaborates with colleagues worldwide, but is here today as a speaker and moderator for Lead Poisoning, a Lifetime Challenge. Dr. Markowitz.
Thank you, Nancy, for introducing our program and for a very comprehensive overview of my career here at Montefiore in the lead program. Dr. Rosen was my mentor before he became my colleague. We had an extremely fruitful relationship in terms of moving the field forward on behalf of lead-poisoned kids and for pregnant and lactating women. So my first role today is to introduce our first speaker, and that's Aaron Rubin, who is a clinical psychologist, an environmental epidemiologist, and a public health science journalist. He's currently completing his training at the Medical University of South Carolina and will receive his PhD in clinical psychology from Duke University next summer. He uses an interdisciplinary research method or research methods to answer fundamental questions about the physical environment and how it may influence brain health across the lifespan from birth to old age. Primarily using cohort studies in the United Kingdom and New Zealand, he's investigated the neuropsychiatric consequences of diverse environmental exposures, including lead, plasticizers, mercury, and air pollution. This work has led to new discoveries about the developmental origins of adult disease, the contribution of heavy metals to adult personality and to psychopathology, and the role of epigenetics in the interplay of the environment and health. His clinical work focuses on child and adult neuropsychologic assessment, as well as therapy for disorders with cognitive complaints, including ADHD, autism, major depression, PTSD, and insomnia. Aaron, the floor or the window is yours.
Thank you so much, Maury. I'm going to just share my screen. Okay, thank you so much. Good morning to everyone. My name is Aaron Rubin. As Maury mentioned, I'm a psychology intern now at Medical University of South Carolina. Thank you for that great introduction. And it's really a pleasure to be here today. So we all know that lead exposure is detrimental to the health and development of children, but what is less clear are the lifespan consequences of early life lead exposures. Today, I'll share with you what we've learned following one population of lead-tested children for 40 years. And before I begin, I'd just like to acknowledge that I'm speaking today from the ancestral land of the Cusco people in coastal South Carolina. So I've organized this talk into 3 stages. First, I'm going to remind you of the history and context of lead exposure in developed countries over the past century. And then I'll describe the specific population we've been following. And then I'll walk you through some of our main findings about what we've learned about lifespan consequences of childhood lead exposures. To begin with, I'll describe the historic trends in lead exposure that we saw in industrialized countries over the past 100 years. I think this is important context to understand our findings. While lead was widely used in paints, pipes, and solder for decades, the greatest exposures really occurred during the era of leaded gasoline. So lead was first added to gasoline in the 1920s to improve engine performance. And this chart shows the consumption of lead in gasoline by year in the United States. Lead's use in gasoline quickly grew through the war years, with strong efforts from manufacturers to motivate consumers to use more leaded gasoline. And this increase continued until the mid-1970s, when the EPA first announced lead and gas restrictions. At that time, we also began population lead level surveillance. And so the red line that I've added to this chart is now going to show you average blood lead levels in American citizens, with the lead level charted on the right side of this graph. And what we saw is that as lead in gas levels declined, so did population blood lead levels. This chart shows the average blood lead level among US children in the late 1970s. The left side is young children and the right side preschool age children. And the dotted red line at the bottom indicates our current level of concern. And I'm showing you this because I want to remind you just how much greater lead levels were during the peak era of leaded gasoline. So what you should notice is that the average child in the late 1970s had lead levels 3 to 4 times greater than what now would trigger clinical concern and case management referral. So at that time, as with today, exposures were borne disproportionately by disadvantaged or minority populations. And in this case, Black children had on average much higher exposures than white children. We've recently determined that this means almost anyone in their 40s now had childhood exposures far in excess of the current reference value. So this chart that I'm showing you is from a paper led by Mike McFarland at Florida State University that we now have under review. And it shows the percent of the current US population that had high lead exposures as children. You'll see the age bands at the bottom. Any age group with the yellow, orange, or red bands had high exposures as children. And these trends were identical across other developed countries. So now I'm charting for you the decline in blood lead levels seen from the 1970s through the 1990s in several English-speaking countries: New Zealand, Canada, the US, the United Kingdom. And what we saw is that as lead and gas levels were removed, we saw population level declines in blood lead levels, so that we really think lead exposures peaked around the world in the late 70s. So we know that lead exposure disrupts brain development, and it appears to do so by substituting for calcium, which is critical in the brain for healthy neuronal processes. And this in turn disrupts neuronal signaling, proliferation, differentiation. And we know that children exposed to lead consequently develop lower cognitive ability, lower fine motor skills, lower emotion regulation capacity. And we know that thanks in large part to the other speakers presenting today and to this great program. What we don't yet fully understand is how these deficits might play out over the lifespan. So my research question and my team's motivating research question has been to investigate the long-term implications of lead exposure, specifically for those individuals born during the peak era leaded gasoline. What we've learned is important for individuals alive today who had high lead exposures in the past, but of course it's also important for today's children who might have high exposures for other reasons, as well as for communities like Flint, Michigan, that were exposed to high lead levels en masse. So if we want to understand the long-term consequences of exposures from the peak era of leaded gasoline, data that can help answer that question can only come from a handful of studies that collected lead measures among children during the era of leaded gasoline. In my combing of the literature, I found 9 such studies. They're largely clustered in North America and Oceania, although there is at least one from Europe. And so far, only 5 of those have received adult follow-up. And I'm now going to show you the ages of the latest follow-ups in each of those study groups. And as you can see, there's been a real range of follow-up durations. But importantly, this has been an international effort to try and understand how early lead exposures play out over the lifespan. And today I'll share the results from our New Zealand studies, which we've now extended up to age 45. Although New Zealand is considered clean and green, you should know that it once had the world's highest levels of lead in its gasoline. And as a consequence, lead exposures owing to vehicle emissions in the past were exactly on par with exposures that were seen in the US, the UK, and other developed countries during the era of leaded gasoline. And this photo at the bottom is from an ad campaign recently rolled out by the country of New Zealand to increase tourism, specifically before the COVID era. Our study takes place in the town of Dunedin, which is a mid-sized city on the coast of the South Island of New Zealand. And we've been following everyone born in 1 year, April 1972 to March 1973, who altogether are representative of the full population of the South Island. And this is a photo of the town of Dunedin, or I should say the city of Dunedin. And you can see it has nice rolling hills and a bay that leads out to the ocean. Dunedin, incidentally, was once a whaling capital of the world. And for a short time in the 80s, it was an epicenter of the global grunge rock scene. There are approximately 1,000 people in our population representative cohort, and we've been continually assessing them every few years for the past 4 decades. This chart lists the dates and ages of each of our assessments, along with the percent of living cohort members who participated in each wave. And as you can see in the column at the far right, retention has been in the mid to high 90s for nearly the entire study, which means our sample has remained population representative at each assessment phase. In the early 1980s, in the early 1980s, when the effects of lead exposure became an international research interest, blood lead measurement was added to the cohort assessment. And we consequently have archival blood lead data for 579 cohort members, who represent the children who were seen at the assessment unit who freely agreed to give blood. And 579 may not sound like a large number, but it actually represents one of the world's largest lead tested cohorts. This scatter plot shows family socioeconomic status by childhood blood lead levels. And the red line indicates the trend between the two variables. And what you can see is that children across the socioeconomic spectrum in our population experienced high lead exposures regardless of social class. And this is unique because in most cohorts around the world, children from poor families tend to experience the highest exposures. And that makes it difficult to disentangle the consequences of lead exposure from all the other consequences that we know accrue from socioeconomic disadvantage. Incidentally, the blue line at the bottom indicates the current reference value. So you can also see that most study members experienced lead exposures that today would have caused clinical concern, but which at the time were considered harmless. So we've benefited in this New Zealand cohort from a population representative sample with high lead exposures, no socioeconomic confounding, and repeated assessments across the first 4 decades of life. And now I hope you're wondering, well, what have we found? And I'm going to walk you through a handful of our findings from long-term follow-up. So we've conducted follow-up studies thus far in 5 broad domains: psychiatric, cognitive, brain structural, socioeconomic, and criminal justice. And because of limited time, I'll only share our findings on the first 4 today, but I'd be very happy to refer anyone to our criminal justice study and to the criminologists who work on our team and are interested in lead exposure consequences for conduct disorder, antisocial behavior, things like that. I'm going to start with our psychiatric findings, as this was one of the first follow-up studies that we conducted. And we did this because we know that children exposed to even low levels of lead tended to demonstrate more behavioral and emotional problems than their peers. And we wanted to know, do these problems extend into adulthood? Do they resolve? Do they develop or change over time? And the findings I'm going to describe were reported by myself and my co-first author, Jonathan Schaefer, who's now at the University of Minnesota, in JAMA Psychiatry in 2019. So again, we tested lead exposure in the Dunedin cohort at age 11, and we then tracked mental health and personality across adulthood. And we did that through interviews at each assessment phase about symptoms of mental illness in the past year. And the interview was structured around DSM diagnostic criteria. And we also assessed personality through surveys of informants who knew our study members well, typically friends or family. In clinical interview, study members were asked about past year symptoms spanning a wide range of different psychiatric disorders, from conduct disorder and depression to OCD, mania, and psychosis. And then we transformed individual symptom counts through factor analysis into a continuous measure of general psychopathology. And we did that because we know that mental illness disorders operate on a spectrum and are not actually discrete categorical entities. There's often high comorbidity at any one time and across time. Researchers have thus found that a higher order factor emerges from symptom counts across domains that we call the P factor of general psychopathology, which is similar to the G factor of general intelligence, which led to the concept of IQ. The construct of P captures the fact that people who have symptoms in one domain of psychiatric disorder tend to also have symptoms in other domains. And in fact, we can determine a person's overall liability to experience mental illness writ large through this continuous factor of psychopathology. And this approach is particularly useful in studies of lead because it allows us to take into account both the severity of illness that an individual might experience and the extent to which they experience comorbidity at one time or across time. So for every participant in the Dunedin study, we've generated a continuous measure of general psychopathology through confirmatory factor analysis of symptom counts assessed at each age and each wave in multiple disorder domains up to age 38 for this particular study. And we wanted to know whether children exposed to more lead develop greater overall psychopathology scores across adulthood. And they did. We found that children with high blood lead levels experienced more psychiatric problems across adulthood. This chart plots the group mean of psychopathology scores across adulthood for study members with increasing levels of blood lead in childhood, binned into groups of 5 micrograms per deciliter of blood lead. Kids on the left of the graph had low exposures, kids on the right had higher exposures. For ease of understanding, I've scaled our psychopathology factor to have a mean of 100 and a standard deviation of 15. And that matches common IQ scales and tends to make more sense to clinicians. So 100 represents the cohort average in terms of mental illness symptomatology. And what we see is that children at the lower end of lead exposure tended to develop fewer symptoms on average across adulthood in the cohort, while kids at the higher end tend to develop more psychiatric illness symptoms than their peers. And in fact, what you'll notice is an overall linear trend of a dose-response relationship. So I'll point out that the effects were pretty moderate. Remember that 15 is the standard deviation for the psychopathology score. There's thus a clear trend, but lead probably doesn't represent a primary cause of mental illness for any one person, although our findings suggest that lead is likely contributing to the experience of mental illness problems. In order to put the size of the effect in context, I've plotted it here against other known risk factors for mental illness, specifically childhood maltreatment and family history of mental illness, both of which are very well described, very robust risk factors for a whole host of psychiatric problems in adulthood. We found that while modest, psychopathology associations with lead are of a similar magnitude to, although smaller than, these well-known risk factors. So the association of lead with psychopathology is about half the size of these other factors. So we can think of the risk following lead exposure as less extreme than risk following childhood maltreatment, such as abuse and neglect. But lead exposure was far more widespread. And in fact, if you'll remember, over half the cohort had blood lead levels 2 times higher than the current reference value. And that was the general trend across the entire developed world. Unlike family history of mental illness, we can modify and reduce children's lead exposure. So I would propose that lead is actually quite a profound contributor to mental illness, not because of the size of the effect, but because of the magnitude of the exposures and the fact that we can modify it. We also measured personality using the Big 5 inventory of personality via informant reports. So we asked people who knew our study members well to describe their personality across 5 important broad domains. And I'll briefly describe these in case you haven't come across them before. Openness at the top describes a person's curiosity or interest in new ideas or experiences and their creativity. Conscientiousness describes a person's tendency to be self-disciplined, controlled, and organized. Extroversion describes a person's interest in engagement with the outside world and energy for social interaction. Agreeableness is a person's interest in social harmony and getting along with others, being kind, considerate, willing to compromise. And neuroticism describes a person's tendency to experience negative emotions like sadness and anger. We wanted to know whether adults exposed to lead as children would show any differences on these broad measures. And they did. So in a second, I'll show you trends on this radar chart, but I just want to orient you to how to interpret it. So we've z-scored our personality measures so that lines following along the 0 axis in the middle represent mean scores across the cohort. Lines further out in the green zone indicate scores that are higher than the cohort mean, and scores closer to the center in the orange zone indicate scores lower than the cohort mean. And now I'm plotting mean personality scores for adults with childhood blood lead levels above and below 10 micrograms per deciliter. So 10 micrograms per deciliter is 2 times the current reference value, which triggers clinical case management, and approximately the mean overall blood lead level for the current reference value. Red here shows personality scores for children above the mean on lead, and blue shows personality scores for children below the mean on lead. We found that across all domains, children with higher blood lead levels were, as adults, more difficult people. They were less conscientious, meaning less organized and demonstrating less self-control. They were less agreeable, meaning they were less likely to get along with others and less likely to be helpful or considerate. And they were slightly more neurotic, meaning they tended to feel negative emotions more than the cohort on average. And I just want to highlight the conscientiousness finding because that was the strongest effect, and because conscientiousness has been robustly associated with a lot of important life outcomes, including educational attainment, occupational attainment, health, wealth, and happiness across adulthood. So being less conscientious, even modestly, can negatively influence your life trajectory. Actually, motivated by our finding, a study group led by Ted Schwaba at the University of Texas recently investigated internet survey data from over 1 million people to determine that those who grew up in counties with higher atmospheric lead levels actually had less adaptive personality profiles. And in fact, individuals born before and after, so individuals born after lead was removed from gasoline tended to be more agreeable, more conscientious, and less neurotic on average than those born during the era of leaded gasoline. And this chart, which I pulled from Ted's paper, shows mean personality profiles in US counties before and after the phase out of leaded gasoline. So this represents a really large replication of our New Zealand findings. So our first takeaway is that childhood lead exposure may raise an individual's risk of developing psychiatric problems in adulthood, alter personality in disadvantageous ways. And now I want to share our next branch of follow-up findings, which are in the domain of cognitive ability. And these studies were motivated by the fact that lead-exposed children tend to develop lower cognitive ability than their peers. And it wasn't clear whether those deficits were maintained over time, whether they changed or worsened. So we want to know, did lead-exposed children experience differences in cognitive ability in adulthood? And those findings that I'm going to share now were published in the Journal of the American Medical Association in 2017. So we measured cognitive ability in childhood before lead assessment, and again in adulthood at age 38 and age 45 using high quality cognitive tests that yield overall IQ scores, specifically the Wechsler tests of child and adult intelligence. And this allowed us to ask whether children exposed to lead experience any deficits in IQ in adulthood. And we found a few things. First, we found that children with higher blood lead levels demonstrated lower overall cognitive ability at age 38. For the moment, I'm not yet showing you our age 45 findings. And this chart shows mean IQ scores at age 38 for children in each of our increasing blood lead level groups. At the left are low exposed children, and at the right are kids with higher exposures. And again, I'm grouping kids into 5-unit increments of blood lead, which is about 1 standard deviation in our group. While the trend isn't as clear as our psychopathology findings, we found that for every 5 micrograms per deciliter higher of blood lead in childhood, adults demonstrated a relative deficit of nearly 2 IQ points. But we also wondered whether there were longitudinal cognitive changes over time, something we could assess because we'd measured IQ in childhood prior to lead measurement. And there were, in fact, longitudinal changes. We found that children with higher blood lead levels tended to experience decline in cognitive ability as they aged. This chart now shows change in IQ from childhood to age 38 by lead group. And now the trend is a lot more clearly linear. What we see is that children with lower exposures actually tended to see their IQs go up slightly over time, while kids in the higher exposure groups lost IQ points. And when I compare the lowest and highest exposure groups, we find a nearly 6-point IQ difference. And that's difference in change in IQ over time. You might find yourself wondering, well, what's a few IQ points here or there? While a few IQ points loss represents a small decline, we found that there were actually meaningful consequences. And I'm not going to talk about our socioeconomic findings in a lot of detail, but I want to mention them now here briefly. When we compared study members at age 38 to their childhood families, study children with the highest lead exposures tended to experience downward social mobility relative to their parents. So now I'm charting social class change from childhood to adulthood by lead group using a 6-point social class scale. And here you can again see a linear dose response relationship where children low in lead tended to see modest upward mobility from childhood to adulthood, while children high in lead tended to see modest downward mobility. Again, we're talking about small differences. 1 point on this scale is roughly a standard deviation in the population. But this trend is fairly compelling when you think of the low lead group as a sort of control group who tended to do better than their parents, which is our goal for society, while the rest of the cohort either stayed the same or went down. When we conducted a mediation test, we learned that decline in IQ accounted for approximately 40% of the effect of lead exposure on socioeconomic mobility. So changes and in fact loss in cognitive ability was a strong driver of this downward social mobility. And lastly, we found that IQ decline has continued across assessments. So now I'm going to tell you about our age 45 findings. Here I've turned those points that I showed you before into bars so that you can better see the differences. And now I'm plotting change in IQ from childhood to adulthood, but I'm showing you decline to age 38 in light blue and decline to age 45 in dark blue. And what you can see is that kids in the low exposure groups actually saw no change in IQ from age 38 to 45, which is what we would expect. 38 is a little too young to anticipate any kind of change in cognitive performance. But what we see is that kids in higher exposure groups actually saw their abilities go down even more than at age 38. And so that decline is continuing and actually expanding over time. And now by age 45, we see an 8-point IQ change difference between our low and high exposure groups. And I anticipate that these differences are going to grow over time. So what are the takeaways from our cognitive findings? First, childhood lead exposure may result in lower overall cognitive ability at midlife. Second, that this represents decline across time. Third, that it results in actual downward social mobility. And lastly, that decline seems to accelerate in the second half of the lifespan. So because of our identified findings of a decline in cognitive ability over time, we're now concerned that children exposed to high lead levels may be at elevated risk for cognitive problems in old age, specifically dementia. And this motivated a follow-up into brain structural differences in midlife. And I'll now share with you our findings in that domain, which I published in the Journal of the American Medical Association last year with my co-first author, Maxwell Elliott, who's now a psychology intern at Harvard University. So remember, we measured blood lead at age 11. And in the last assessment phase of the Dunedin study at age 45, study members participated in high-resolution structural magnetic resonance imaging to give us detailed pictures of the overall integrity of their brain tissue at midlife. And for this follow-up study, we considered 3 related domains of brain tissue. First, gray matter, which is the outer layer that contains most of the brain's neuronal cell bodies and is involved in higher order processes like speech and decision-making. Second, white matter, which is deeper in the brain and contains most of the brain's myelinated axons that extend from neurons for long-range communication across brain regions. And third, an overall estimated brain age, which is based on algorithms trained in other data sets with diverse ages to predict someone's age based on how their brains look. And that gives us a global overall impression of the aging of the brain. And I'll just show you briefly the gray and white matter differences a little more finely. So in this figure, you can see their neuronal bodies in the gray matter band at the left, and their long axons extending in the white matter at the right. And then this figure describes some of the anatomic differences that can differentiate young and old brains, including things like atrophy across the gray and white matter, wider vasculatures reflecting, again, atrophy, and potentially lesions, particularly around the vasculature, which are those kind of bright white spots. So what did we find? First, we found that children with higher lead burden had lower gray matter integrity at age 45. This scatter plot shows the association of blood lead levels in childhood with the brain's surface area 4 decades later. And surface area captures things like the number, size, and density of neurons. So the effects here are modest, but in general, brain surface area was lower among adults with higher lead levels 3 decades earlier. And second, we found lower white matter integrity. This is a scatter plot showing the relationship between blood lead levels and the brain's fractional anisotrophy, which is a measure of how tightly organized diffusion of water molecules is across the brain. So lower anisotrophy means diffusion is unrestricted, and higher anisotrophy means there's tighter restriction on water diffusion, and that's an overall measure of the size, density, and quality of myelinated white matter in the brain, because more robust white matter restricts the flow of water molecules along the tracks. And we found that children with greater lead exposure by midlife were adults that had modestly lower fractional anisotrophy, meaning lower integrity white matter across the brain. And third, we found that higher blood lead levels in childhood associated with older looking brains at midlife. So here I'm plotting what we call the brain age score against childhood blood lead levels. A brain age of 0 would suggest that a person's brain age matches their chronological age perfectly. So if you're 45, your brain looks like the brain of a 45-year-old. Higher scores on the brain age indicate brains that appear older than a person's age, suggesting that they may have experienced accelerated brain aging. including some of the differences we discussed before. So atrophy, lesions, expanded vasculature. What we found is that adults with higher childhood lead burden had older looking brains by midlife. And age 45 is not that old. We were actually surprised to find such a wide variation in brain age scores, and that lead exposures decades before predicted brain age. So adults with higher lead burden tended to have brains that looked 1 to 2 years older than their peers when we look at the higher and lower exposure groups. So what are the takeaways from our brain findings? Childhood lead exposure may result in lower overall brain integrity at midlife and accelerated brain aging. And I want to put a little caution around those findings because the measures I just described are cross-sectional, meaning we've only assessed them at one time point. So we don't know if the differences we detected represent change over time or are simply childhood consequences of disrupted development that have maintained for 40 years. So if we'd had the benefit of taking brain images when our kids were growing up, we would know, are the differences we're seeing truly, for example, atrophy, truly accelerate aging, or do they just look older because their brains were damaged early on? Now, I suspect that what we're seeing represents true change over time because we've seen cognitive changes, and cognitive ability is a really fine and sensitive measure of the brain's integrity in a way more so than structural, anatomical imaging. But that's a hypothesis that we're going to have to assess. And again, most people in their 40s had high exposures as children. So if there are implications for brain aging and dementia risk, now is an important time to understand whether we would need to intervene with adults who were exposed to lead as children. And that's a number of hypotheses that we plan to test at Dunedin Study's next assessment phase, which is going to begin in 2024. So what are our overall takeaways? First, childhood lead exposure really seems to have long-term, multi-decade ramifications, and individuals do not fundamentally seem to recover from early harm, at least in this population where there was no intervention, because the exposures that our study children experienced were considered at the time to be harmless. Second, individuals may experience greater psychological distress and personality difficulties across adulthood. Third, individuals may experience decline in cognitive ability over time. And fourth, that those declines might be accompanied by downward social mobility. Again, modest downward social mobility, but downward social mobility nonetheless. And lastly, that brain structural differences suggestive of accelerated brain aging are apparent by midlife. And my overall conclusion that I want everyone to take away is that childhood lead exposure can influence the way people think, feel, and behave across the lifespan for decades to come. So I'll end now in order to have time for questions. But I just want to thank my advisors for introducing me to this topic, as well as for guiding and shaping this research every step of the way. And that's Temi Muffet and Avshalom Caspi at Duke University, and the full Muffet-Caspi lab group, including our many co-authors and collaborators, only a few of whom are listed here. And this photo on the right shows a few of us enjoying a meal in Dunedin at the house of the director of the Dunedin study, Richie Poulton. And this was long before COVID hit. And you can see those hills in the background and the bay that I showed you earlier. Dunedin's really a lovely place to visit. And I'd like to thank the full Dunedin study team. So pictured here is the assessment unit, which is built just for the Dunedin cohort and the ongoing assessments. And this is the full study team at the bottom. So what you can see is that our studies really represent a massive group and international effort. And we're investigating lead, but there are other principal investigators looking at a whole host of other differences across the lifespan. So it's really multidisciplinary research focus. And I'd like to thank our funders at the US, at the National Institute on Aging and the National Institute on Environmental Health Sciences, and in the UK at the Economic and Social Research Council and the Medical Research Council. And we do have funds provided in New Zealand as well. So I'll just end with this slide, and I'll stop sharing in case we want to have some Q&A. Thank you very much.
Thanks so much, Aaron. Fascinating talk. I'm looking at the Q&A right now. I see there is a question in there, but I'm going to jump in and ask a question first. Actually, I have three, but the first is the scores that you showed us, I assume are all in multivariate analyses, so controlled for lots of covariates.
That's right, yeah. We can talk about the kinds of adjustments we made, but in all the tests I showed, we took into account things that we'll typically want to take into account when we're looking at the outcomes. So when we were looking at cognitive ability, we adjust for maternal IQ, for example, which is a good member of sort of genetic propensity towards IQ. For the socioeconomic status studies, we did that as well. We took into account family social class, sex, things like that.
Mm-hmm. A second, also relatively short question is, the Cincinnati group in their cohort did MRI studies when their kids were already in their 20s and found differences in the size of the prefrontal cortex and I think in the hippocampal areas in males, not so much in females. So in your study, it looks like you could do the same kind of focusing in those particular areas to look at the size, to see if you could actually find the same thing or whether it's worse for people already in their 30s or at 45.
Yeah, it's a good idea. We did do these sort of sub-region analyses. So what I showed you today were global findings. In general, to be honest, we didn't see any particular region that seemed particularly vulnerable. What we saw were differences across the brain, and that might have been the result of 20 extra years of aging compared to the Cincinnati cohort. But you're motivating me to look back at our findings and see if there are any notable frontal lobe parietal changes or differences, and whether our effect sizes seem to be similar to what they reported in the Cincinnati study, or if they seem to have magnified over time. It's hard to make comparisons because the lead exposures may have been different, the populations are otherwise different, but it's an interesting idea that we should take a look at.
Mm-hmm. So I'm going to go to the Q&A, and if there's time, I'm going to come in with another kind of a more zinger question.
Please.
So first from Jean Sales Shaw. As lead control workers, we explicitly emphasize the oral pathway of exposure. Aaron, your studies looked at the airway exposure from gasoline use. Is there a basis for airway exposure? And if so, is there today any analogy evidence for air-based lead exposure?
Yeah, so unfortunately, we don't have good purchase on the pathways of exposure. We believe that the lead levels seen in our cohort were the result of lead in gasoline. So there's an atmospheric lead component, but we don't know, were kids inhaling dusts or soil dust with lead in it, which is plausible. We don't know if there was some ingestion. We do know, for example, that a few study members had high exposures due to leaded paint. Those study members actually didn't make it into the findings that I showed today because their lead exposures were really high and they actually didn't survive till adulthood. And I don't know the extent to which lead contributed to that early mortality, but all we know is what the body burden of lead was at age 11. And we don't know what the routes of exposure were. We believe they were likely inhalation of leaded dust, but we can't tease apart what the mechanisms were for adsorbing and generating that body lead burden.
It actually raises kind of an interesting thought that as the lead is coming out the tailpipe, the particle size must vary, and it could be extremely small potentially. So
Yes.
it makes me wonder whether you can have direct absorption through the nose, through the cribriform plate directly into the brain from that leaded gasoline usage, which might have a different impact than on directly on the brain.
That's right. So that, from my conversations with folks who are studying the transduction of air pollutant particles, across the nasal nerve into the brain, there's still a lot of disagreements about how significant that route is for, particle deposition in the brain. From the studies I've seen, for example, in dogs raised in ambient Mexico City air, where you can actually slice the brain at different ages, it seems that small particles enriched with heavy metals very much can reach the brain directly through the nose. And we know that particles that reach the brain directly are going to have not just a direct influence on neuronal death, but then a cascade of changes for the microglia, et cetera. So I agree with you that I think there's greater disruption when particles go direct. What we don't know is to what extent the lead emitted from tailpipes went to the brain directly or circulated in the blood system, because it can reach the blood through the lungs pretty easily.
Mm-hmm. Good, thank you. A colleague of mine from upstate New York, Jennifer Becker, writes, "Thank you, so interesting. Are there regular timeframes for follow-up on the Dunedin group? What is the next stage and what's the long-term plan?"
I love that question because that's exactly what we're gearing up for now. So we've varied, I say we, this is a study that's been going on long before I was born, but the assessment timing has been varied to be developmentally appropriate. So when the study members were kids, they were brought back, within 2-3 years of each other. And then over young adulthood, those were spaced out. Now that they're aging, we're starting to bring them back more regularly. So the last assessment was age 45. The next one's going to be age 52, which is going to start April of 2024, which sounds far away, but we're actually writing grants now so that we'll have money in hand to do all the work we want to do in 2024. So my plan is to submit a grant later this year to try to gather new lead exposure measures, looking at bone lead, looking at blood lead, to try and understand brain changes over time, current exposures, epigenetic differences. I'm going to make this a bigger part of our work in 2024.
And the next question in the Q&A from Teresa Sicalo, I think, or Sicalo, I'm sorry for the mispronunciation. Would you think that people born in the 60s, the 1960s, could have the same results due to their lead exposure to lead gasoline through childhood and their adolescent years?
Same results as what I showed. I mean,
well, an older cohort, so.
Yeah. I would say what we've shown you today are differences in outcomes along a gradient of exposure. So anyone born at any time with a certain exposure could be predicted to have the same outcomes that I showed you. My read of the literature is that folks born in the 60s tended to have higher lead exposures than people born today. And they were on the magnitude of kind of late 80s, early 90s kids. So that my childhood exposures probably look like someone who was born in the 60s, which are still higher than they should be, higher than what we believe is safe or harmless, which is no lead exposure. And then, of course, there's going to be individual differences in your outcomes. So we talked about how lead exposure in early life seems to lead to a decline in cognitive ability over time. Well, if you were already going to be on the cusp of a cognitive impairment for any other reason, you might enter a cognitively impaired state much sooner because you were exposed to lead earlier, regardless of what year you were born. And the same is true for psychiatric problems. People with a high genetic loading for psychiatric risk, let's say they have a high family history of mental illness, they might be at risk for psychological problems without lead exposure, but small exposures might push them into an impaired zone earlier. So I don't, I think about cohort effects, but I really think about what's the exposure been? How consistent has it been? Has there been any intervention? We're working now with surveys in Flint, Michigan to see to what extent mental health problems have persisted after the water crisis and to what extent intervention services have made a difference. And they seem to be making a difference.
There are two other questions. One is, you think that wearing masks due to COVID will have an effect on nasal lead absorption, basically, right?
I love that question. I'll speculate wildly and say, sure, I don't know. I think that's a really good empirical question that we can test. It's a hard question to test because if you want to know what's happening in the brain, the best models are animal models. And I don't know what an animal model for a COVID mask looks like. But we could, you could imagine a study where you see to what extent particles reach the mouth and brain. But we know that masks are good, for example, at blocking and disrupting viral loads into the nose. So will they also disrupt pollutant particles? I would guess so. And that, if you're going to go for a jog in Beijing during a higher pollutant level day, I would wear a mask for a variety of reasons, right? Cardiorespiratory. But in the back of my mind, I do think about brain health, which, incidentally, I've started working more on air pollution exposures today in places where lead is not enriched in the airways. But there's lots of other pollutants that seem to have a similar effect on brain health at the different points in the lifespan and of a similar magnitude. So we did a study in the UK recently looking at psychiatric outcomes of early life air pollution exposure. And they were similar magnitude to the lead findings I just shared with you.
Mm-hmm. Right. So that would be a potential confounder in your own study, since there was pollution varying during that time. The last question in the Q&A is about, is focusing at the low end, where you used 5 microgram per deciliter increments for your bands, but the CDC has changed its reference level 2 weeks ago from 5 down to 3.5, which is epidemiologic data. I don't know that's an impetus to reanalyze all of your data to change that lower band to 0 to 3.5 and then looking above it, because that's not a toxicologic threshold from the CDC in any case.
Yeah, and I'll just point out that I showed you the results in 5 microgram per deciliter bands just for demonstration purposes. All of our analyses used lead continuously measured. So the changes in reference values wouldn't change any of our findings.
Mm-hmm. So staying at that end of the spectrum, I have the following question. In that 0 to 5 group, you showed higher IQs, including the change in IQs, better sized brains for their age. Is lead a positive at low levels?
Yeah, I'm glad you mentioned that. In a way, it would suggest that, somehow they're doing better because of it. I'll really remind everyone that in this group, which is totally representative of everyone born in the 70s in different developed countries, there were almost no truly low lead exposures. I think 94% of our cohort had exposures above 5. And the analyses I'm presenting are really about differences within, representative populations, so that kids with lower exposures did better than their peers on average and much better than their peers with high exposures. I don't know how well they would have done if they'd had no exposure at all, if that makes sense. But what we know is that in New Zealand across the last 50 years, social policies, the economy, things are working out so that kids are doing better on average than their parents. Their IQs are going up slightly. That tends to be a result of education. but that those benefits seem to be confined to kids who hadn't had high lead burdens. Is that a helpful answer?
Yeah, I think so. So we've run out of this portion of the program's time. Thanks so much, Aaron, for really a spectacular talk. Thank you.
Thank you. Real pleasure.
So I'm going to now introduce our next speaker. My pleasure, Bruce Lamphere, Dr. Bruce Lamphere, MD, MPH, who is a professor at Simon Fraser University. I hope that's pronounced right, Fraser or Fraser. And he's an adjunct professor at Dartmouth College. He's led key studies that have been used by federal agencies to set standards for lead in house dust and air, and to conclude that there is no safe amount of lead. He's evaluated the impact of toxic chemicals such as lead, but also phthalates, air pollution, and fluoride on cognitive deficits and behavioral problems in cohorts in the United States and Canada. He's also conducted numerous randomized controlled trials to examine ways to reduce exposure and to prevent disabilities linked with pollutants and toxic chemicals. Dr. Lamphere also produces videos to show how our health is inextricably linked with the environment and to elevate efforts to prevent disease. So I look forward to seeing what new videos you've incorporated in this presentation, Bruce.
All right, well, thank you, Maury, and it's wonderful to be back. I don't know if this is my third or fourth time, but it's always a pleasure to participate in your annual seminars. So let me just get things set up here a little bit. Move some things out of the way. So one of the things that Aaron mentioned is that they didn't look at death, and this next talk will be about death, particularly from coronary heart disease. And coronary heart disease and atherosclerosis is not only the leading cause of death in the United States, but also worldwide. And so most of my work has really focused on children, particularly looking at low levels and the impact on brain development. But increasingly, as there's been a focus on the global burden of disease, what causes the most death, for example, I became somewhat frustrated that nobody really was asking questions about lead. Hypertension, it was said, was a leading cause of death, but nobody was asking, well, what caused hypertension? Or looked at a different way, air pollution was, but had anybody tried to tease out the impact from lead exposure? So after spending several years trying to convince other people who did study mortality, I decided to do a study, and that's what I'll talk about today, and try to put it into a perspective or into a narrative, really, to try to help explain what now has been coined the mysterious decline in coronary heart disease. So 100 or so years ago, Dr. William Osler, who was a Canadian who then became the first chief of medicine at Johns Hopkins and then went on to Oxford, wrote probably the most influential medical text of the time, beginning in 1892, and this one was from 1912, where he said, angina is a rare disease in hospital, angina meaning angina pectoris, pain in the muscle of the heart, oftentimes would predict heart attacks and was an indicator of coronary heart disease and atherosclerosis. One case a month, he said, is the average, even in larger metropolitan hospitals. So like most of the chronic diseases that kill us today, lung cancer, coronary heart disease, chronic lung disease, diabetes, coronary heart disease is a man-made disease. And that's both somewhat tragic, that we fouled our own nest and caused our own demise, but hopeful in the sense that if we cause these diseases, we can cure them. We can actually prevent them. We can't actually cure them very well, but we could prevent them. And so over the last century, what you can see is first, heart disease began to rise beginning in the early 1900s. By 1930s, became the leading cause of death as tuberculosis deaths declined. And it peaked somewhere in the 1960s. And as coronary heart disease deaths were measured separately from heart disease overall, you could see that the peak in deaths from coronary heart disease was in the United States, somewhere in the 1960s, even perhaps closer to 1967 or 1968. And then you can see here the decline in coronary heart disease, as well as overall heart disease. Now, coronary heart disease is still the leading cause of death in the United States and worldwide, but it has declined since its peak in the 1960s by 70%. Now, early on in the 1970s, there was a physician in California who recognized that coronary heart disease was declining. And by the mid 1970s, it was clear that this was a national phenomenon. And so the National Heart Institute, which became the National Heart, Lung, and Blood Institute that we know today, convened a conference of over 80 experts from around the country, which became known as the Decline Conference. So just over 40 years ago, they had this conference, and the purpose of the conference was to consider whether the greater than 20% decline in coronary heart disease mortality or deaths since 1968 was real, to discuss possible causes, and finally to recommend further studies to elucidate those causes. They ended up first saying that the decline was real and that the 20% estimate was reasonably accurate, which meant that 190,000 fewer people had died from coronary heart disease than expected based upon the peak in the 1960s. And they also came up with a whole host of possible reasons for this decline. But as the director of the National Heart Institute, Dr. Robert Levy, said, it should be evident to the reader that we have too many rather than too few possible explanations for the decline. Still, he was quite confident or bold in saying that the decline in coronary heart disease epidemic in the 1970s strongly suggests that we can control and eventually eradicate modern civilized man's most serious killer disease, which I found, looking at it from today, to be extraordinarily refreshing, that here we have a leader from the National Institute of Health who is actually talking about controlling and eradicating something like coronary heart disease, as opposed to what the current focus is on. Let's find new drugs or CRISPR or whatever else to treat disease once it happens, but very little investment and very little talk about actually preventing disease. Now in Britain, Jeffrey Rose, who's become one of my public health heroes, was also quite convinced that this decline in deaths from coronary heart disease, and just to mention that I'll use coronary heart disease and ischemic heart disease interchangeably, they are synonyms, that the decline in US men was real. But then he asked, why is it that in Britain, we're failing to prevent a preventable disease? We'll come back to this later. For those of you who don't know Jeffrey Rose or some of his work, two of his classic articles, including the one I just mentioned in 1981, were published in a book called Strategy for Preventive Medicine in 1993. And I don't know what the other top 10 books are to read if you're interested in public health or population health, but this is one of them, the Strategy for Preventive Medicine. And many of the videos that Maury mentioned are based on some of the concepts that Jeffrey Rose talked about. So these risk factors are all quite familiar. This litany of risk factors is familiar. Male sex, although that's not quite as prominent today, obesity, smoking, diabetes, salt intake, physical activity, and genetic susceptibility. And quite conveniently, these all kind of point the finger at the individual You might even say blames the individuals for their own problems. But they fail to recognize things that are most often outside of the control of individuals, including toxic metals, lead, arsenic, and cadmium, or airborne particles. So I'll be focusing mostly on the toxic metals and to some extent air particles as well. This was a study that was published in 2018, a meta-analysis of 37 unique studies, 26 cohort studies, and 11 case control studies with almost 350,000 non-overlapping participants. And what Chowdhury and others found is, let's focus on lead for a moment, that higher lead exposure was associated with a significant increase in cardiovascular disease, coronary heart disease, and stroke. Now, none of this was a surprise. There were a number of studies, particularly during the 1980s, that showed that as lead exposure increased, the hypertension and heart attacks or coronary heart disease also increased. So there's quite a history of recognizing lead as a risk factor for coronary heart disease and heart attacks and hypertension. In fact, it's an established risk factor, but somehow it never really made it onto that familiar litany of lists, that somehow it was more convenient just to point the finger at people for smoking or for being sedentary. So what do we know about lead and cardiovascular disease? First, lead enhances atherosclerosis in laboratory studies by inactivating nitric oxide, inhibiting endothelial repair, that is the lining on the inside of blood vessels. It impairs angiogenesis, that is new growth of blood vessels, and it promotes thrombosis or clots. These are the hallmark of atherosclerosis and coronary heart disease. So we know that lead in a laboratory setting sends off a cascade that leads to, in laboratory animals, atherosclerosis, hypertension, and the mechanisms underlying coronary heart disease. And yet, despite this long history, no studies had calculated the number of cardiovascular disease deaths in the United States attributable to lead, or anywhere else for that matter. And it was unknown if blood lead levels less than 5 microgram per deciliter or 50 parts per billion, which are typical for adults, are linked with cardiovascular disease deaths. I'm going to use interchangeably microgram per deciliter and parts per billion. All you really need to do is just to add a 0 for lead. It doesn't work like that for other chemicals, all other chemicals, but just add a 0. So 1 microgram per deciliter would be 10 parts per billion, or 10 microgram per liter. Why we use deciliter, maybe Maury can tell us in the question and answers. I couldn't figure it out, other than the fact that lead circulated at much higher concentrations than other metals like mercury and arsenic. Mercury and arsenic we use microgram per liter instead of microgram per deciliter. I think there's value in looking at parts per billion. First, it's easier to try to translate that or communicate parts per billion. 2 parts per billion, I'm sorry, two tablespoons of lead in an Olympic-sized swimming pool is equal to 1 part per billion. So you can kind of get a sense that these are very small concentrations, but still consequential. So what we wanted to focus on is what happens even at these low levels that right now are considered safe or acceptable for adults. We don't accept them for children, but adults, we believe today still don't have any adverse consequences until there's higher levels, or at least we don't act as though they do. So I want to spend the next few slides just talking about a study we did using the National Health and Nutrition Examination Survey. And we were able to follow up over 14,000 adults for a median of 19 years. Of those, about 4,400 participants died. 1,800 or 38% due to cardiovascular disease, almost 1,000 or 22% due to ischemic heart disease or coronary heart disease. The geometric mean blood lead levels of the participants was 2.7, and about 20% had a blood lead concentration greater than 5 microgram per deciliter, or 50 parts per billion. Not surprisingly, we found that participants who had higher blood lead levels were older, less educated, and more likely to be male. They were more likely to smoke, consume larger amounts of alcohol, have less healthy diets, high serum cholesterol, higher rates of hypertension, and diabetes. This is true for any kind of risk factor that we might want to study. So when we've looked at smoking, for example, and coronary heart disease, in theory, we should adjust for all of these factors. Rarely, however, have smoking studies adjusted for lead when we've looked at coronary heart disease, and certainly not tried to estimate the burden of disease, the number of deaths in a given country. So here we can see the hazard ratios, which is comparable to a risk ratio for all-cause mortality, that is any type of death, and cardiovascular disease mortality. And cardiovascular disease mortality encompasses coronary heart disease or ischemic heart disease. So first we can look at all-cause mortality, that is any type of death, and we can see that there's about a 40% increase in any type of death, premature death if you prefer, associated with higher blood lead levels. And this is looking at an increase in the continuous blood lead levels from the 10th to the 90th percentile. So we're trying to look really the entire, almost the entire continuum, but not let the tails dictate what we see. And you can also see that we've adjusted for a whole host of different factors. And in fact, NHANES 3, which was our baseline data, was specifically set up, or one of the key things it was set up to answer questions about was the coronary heart disease epidemic. So for all types of cardiovascular disease, including ischemic heart disease, we could see about a 70% increase in the hazard ratio, and for ischemic heart disease deaths, twofold increase. The first thing we wanted to look at beyond all deaths was to look at what happened in the participants who had a baseline blood lead less than 5 microgram per deciliter. And you can see here, it's very similar to what we saw before. For all-cause mortality or all types of death, about 38% increase. For cardiovascular disease mortality, almost a twofold increase. And for ischemic heart disease mortality, over twofold increase. So now we can look at the shape of the dose-response relationship. And this is really important and not really thought of enough when we think of exposures to toxic chemicals. 50 years ago or more, when regulations were being developed for toxic chemicals, at least for non-carcinogens, the assumption was made that there is a safe level or threshold below which no harm occurs. Now that was very convenient for the chemical industry, and in particular for the lead industry, because this was really promoted by Robert Kehoe, who was a professor at the University of Cincinnati, as well as the medical director for the Tetraethyl Lead Corporation. That this argument was, of course, there's safe levels. We don't have to worry about background exposures to the population. But what you can see here is that as the concentration of lead in blood increases from 0 to 2.5 and 5 and 7.5, that the hazard ratio increases. But in particular, what's worth noting is that the increase starts immediately at the lowest measurable levels, which was 1 microgram per deciliter when NHANES was done. That was the detectable level at the time. And you can see that the risk increases rapidly beginning at the lowest measurable levels. So what does that mean? Well, first it means that there is no safe level, which is in contrast to the way we treat lead exposure in adults and how we regulate most chemicals today. And so if there is no safe level, then one of the things that Aaron alluded to becomes extraordinarily important. Not only do we need to think about the risk ratio or the hazard ratio in this case, but the proportion of the population that's exposed. And essentially, everybody's exposed. There are no unexposed people, and that's still true today. We all have measurable levels of lead in our blood and our soft tissues. So just to make that more clear, if we look at something like ischemic heart disease or coronary heart disease mortality, we see the same thing. It's actually a bit steeper. The hazard ratios for smoking is actually larger than what we're seeing here for lead. But the difference is only about 20% of people smoked. And so the overall population impact was much greater for coronary heart disease from lead exposure than it was from smoking. So here, looking at coronary heart disease, you see basically the same pattern. No safe level, steep rise in the risk beginning at the lowest measurable levels. All right, so what does that mean? So we can look at the attributable fraction, that is the estimated number of deaths due to lead exposure after taking all these other factors into account. When we look at all-cause mortality, we estimated that there were about 400,000 avoidable deaths every year in the United States. That makes lead the third leading cause of death. It also makes it the leading cause of the leading risk factor for premature death from coronary heart disease. Excuse me. Let me get down to that in a minute. So all-cause mortality, we estimated over 400,000 avoidable deaths. Now that's kind of non-specific. We don't try to dig in and say, well, are every one of those deaths specifically linked back to lead as a risk factor? So let's focus on the next two, cardiovascular disease mortality and ischemic heart disease mortality. For all types of cardiovascular disease mortality, we saw about a 28% attributable fraction. That is, we estimated that about 28% of deaths from cardiovascular disease were the result of lead exposure, 250,000 deaths every year. And for coronary heart disease mortality, almost 40%, or 185,000 deaths from coronary heart disease were from lead exposure. That makes lead the leading cause of dying from coronary heart disease. And so, as Maury mentioned, I like to make videos. My brother is a graphic designer, and we take time to try to take some of the scientific information that's out there and help translate it to make it more easily understood by large groups of people. So I want to show this, and remember again that 5 microgram per deciliter is equivalent to 50 parts per billion.
Lead is also a risk factor for coronary heart disease. That's when your heart is slowly or suddenly suffocated by an obstruction of the arteries feeding the heart. In a national study, we found that deaths increased sharply at blood lead levels below 50 parts per billion, the level considered safe. There was no threshold or safe level. If we let the narrowing of this coronary artery represent the risk of dying from heart disease, you can see that most of the narrowing occurs below 50 parts per billion. We found that lead accounts for about 185,000 deaths every year. That makes lead the leading cause of fatal heart attacks in the United States.
Oops, sorry about that. So now that we've looked at the study that shows that lead's much more consequential than most of us realize for coronary heart disease, let's go back now and try to look at this rise and decline of coronary heart disease and see if we can try to understand why it happened. So first, I want to go back to Jeffrey Rose's study or commentary, actually, in 1981, where he acknowledged that the decline in deaths from coronary heart disease in the United States appeared to be real, but it didn't happen in Britain. Now, Jeffrey Rose didn't really appreciate the full impact of air pollution or lead. but we can compare what happened in the United States with blood lead levels. And these blood lead levels were based upon some unpublished work by Rick Nevin. And some of you may know Rick from his work on lead and trends in criminal behavior around the world. And he was able to calculate these based upon air lead levels and blood lead surveys that were done, in this case, in the United States and in the next slide in Britain. And so you can see here what I already showed you first in blue, that deaths from coronary heart disease in men had begun to decline in the 1970s as blood lead levels declined as well. So why didn't they decline in Britain? Well, here you can see for British men, no change, maybe a little bit going on here, but no real change until the mid-1980s, nor was there much of a change in blood lead concentrations. Air pollution came down quite dramatically beginning in the 1970s in Britain. They had a lot of work going on with control of air pollution, but lead wasn't taken out of petrol until the mid-1980s. And so that's when you begin all of a sudden to see a decline in coronary heart disease. We can look more specifically at hypertension. And I think I already mentioned that lead is an established risk factor for hypertension. That's not a question. But what we did here was, when I was trying to go back and understand the history of lead exposure and coronary heart disease, I was familiar with lead, but I wasn't so much with coronary heart disease. I found this precipitous decline in hypertension in American adults during the 1980s. You can see here from 1976 to 1980, that's the solid blue lines, that during that time period, this is with NHANES 2, about 1 in 3 adults, 1 in 3 American adults, that is over 20 years of age, met the definition of hypertension. 1 in 3. This is striking. What's even more striking is if you go out to NHANES 3, and in this case, 1988 to 1994, is that a decade later, only 1 in 5 US adults had hypertension. Now, other epidemiologists looked to try to understand whether that was because of changes in smoking. No. Changes in treatment for hypertension? No. Changes in obesity or BMI? No. In fact, this is the decade where obesity really took off. So the opposite happened. As people became heavier, they actually had to use a different size blood pressure cuff in these studies. And so they made sure that that wasn't an artifact, and it wasn't. And so what you can see here is this precipitous and unexplained decline, huge decline in hypertension. So some of you who've been familiar with the lead now won't be surprised to know that what did correlate strongly with this was the decline in blood lead concentrations, because lead was taken out of gasoline. And so you can see almost in lockstep this decline. And it almost seems like year to year, the changes are evident. So now we have some evidence that over the past, there's been this decline in hypertension as blood lead levels plummeted when lead was taken out of gasoline. We can also look at a different time period. This is 1988 to 2004, also taking advantage of NHANES, but at a later date than what I just mentioned. And here, Adrian Ruiz Hernandez and Ana Navas-Asian and their team did a much more sophisticated analysis. The one I just showed you was unadjusted. And they found that about 32% of the ongoing decline in cardiovascular mortality in the United States during this time period was due to reductions in lead and cadmium. Overall, about 18% was due to lead. So they're seeing as a very similar pattern with the ongoing decline in lead exposure and coronary heart disease deaths. Now, there were, of course, other things that were going on this time that we need to think about. Just like with lead and coronary heart disease, we know that as air pollution increases, you see premature mortality from all causes, as well as coronary heart disease. And the shape of that, in this case, exposure response, because we don't have a biomarker for air pollution, we have to rely on external measures like air monitors. But you can see that there's a similar pattern. That is a very steep increase in the risk of dying prematurely from all-cause mortality. And several studies also show specifically for cardiovascular disease mortality or coronary heart disease, again, with no evidence of a threshold or safe level. And in fact, despite that assumption I shared with you a few minutes ago, that most of the non-carcinogens, virtually all of the non-carcinogens, are assumed to have a safe level of threshold. What we know from lead, air pollution, benzene, asbestos, smoking, is that there is no safe level of threshold, and yet that's how we regulate toxic chemicals. And that means we're underestimating their impact, and that means we're failing to protect the public from these widespread exposures to toxic chemicals. So we have to think about air pollution as well, and we have some insight from Britain. As I said, air pollution did decline during the 1970s in Britain, but they didn't see that steep decline in coronary heart disease deaths until lead was taken out of gasoline. Now, my guess, because we don't have good data that incorporates both lead and air pollution, is that they both contribute, but that lead appears to be more prominent than air pollution, or has a larger risk than air pollution exposure, but that still is yet to be determined. We do have some insight on levels of air pollution from secondhand smoke. So this was a study, and it's been replicated in other countries, but this study in Scotland was particularly informative because they were able to look at what happens to heart attacks following the ban on smoking in public places among, this is the key part, among non-smoking adults. So what they found was there was a 20% reduction in heart attacks among non-smoking adults following the ban on smoking in public places. That's just from reducing community levels of secondhand smoke, a 20% reduction. Now that fits if we go and look at a study that Arden Pope had published in 2009, and he looked at the adjusted relative risk here of coronary heart disease by PM2.5, by airborne particles less than 2.5 microns in diameter. Again, a very steep risk beginning at the very lowest levels. Now, he showed that for particulate matter, but he also looked at it in relationship to the average number of cigarettes. This area here, though, will represent secondhand smoke. That point where maybe even just a decade or more ago we thought of as inconsequential, but you see already a very steep increase beginning at the lowest measurable levels. And so now we can begin to put all of this together a bit more. Mass manufacturing of cigarettes began here. We had a lot of coal, but then the automobile began to really take off here. And then in 1925, lead was added to petrol in the United States. So what can we say about some of these trends that might help us even have a bit more confidence that this connection between lead and coronary heart disease is real? Well, we can go back and look at a study that Norm Robbins did in Cleveland. And what he did is he looked at the teeth of men of various ages, as well as the sediment, lake sediment, and showed that the amount of lead in the teeth varied over time and represented exactly what I think most of us expected. And that is the peak of the lead pandemic was indeed during the 1960s. And you can see it increased here very much in line with this pattern of coronary heart disease deaths, beginning first, of course, with the overall surveillance for heart disease, and then more specifically for coronary heart disease. And some of this shouldn't actually surprise us, because even back in the 1880s, Dr. Lorimer in Edinburgh found in his case series of men with saturnine gout, that is lead-induced gout, that he saw a very consistent pattern of arterial thickening and degeneration, that is arteriosclerosis or atherosclerosis in these 107 cases. So there is a large body of evidence that began back during this time. In 1925, Yandell Henderson, who with Alice Hamilton was part of a blue ribbon panel that argued that we should not be adding lead to gasoline, said that it will lead to a scourge worse than tuberculosis, with massive hardening of the arteries. And so we've had a warning about this, but still conveniently found ways to ignore it. Now, we can also look at trends in cigarette consumption, and that too very much fits this pattern of the rise and decline of heart disease and coronary heart disease. And in the first Surgeon General's report, already they identified that smoking was a likely risk factor for coronary heart disease. Now, what does that mean? Well, first of all, it's important to note that smoking is really a whole cocktail of different chemicals and particulates, including lead and cadmium and airborne particles. It's not one thing. And so, for example, if you look at smokers, on average, they have blood lead levels that are about 20% higher than non-smokers. And non-smokers who are exposed to secondhand smoke have blood lead concentrations that are about 20% higher than non-smoking and unexposed people. So we really can't separate out airborne particles and metals from smoking and secondhand smoke. They all seem to be connected. So what can we conclude from all of this? Well, we can't say that lead alone caused the rise and decline, this mysterious rise and decline in coronary heart disease deaths. But I think we can conclude that heavy metals and air pollution, or heavy metals in air pollution, were the mysterious culprits of the rise and decline in coronary heart disease. And yet, strangely enough, most people have no idea that lead, or even air pollution for that matter, or secondhand smoke is so consequential for this leading cause of death and disability around the world. Now, once we acknowledge that lead and leaded air pollution is a predominant or prominent risk factor for coronary heart disease deaths, we can also begin to understand other persistent mysteries. First of all, why was coronary heart disease mortality higher among city dwellers in the 1970s? They were exposed to higher levels of lead and air pollution. Second, why is acidic water a risk factor for coronary heart disease mortality? Because acidic water would leach lead from lead service lines and make it more bioavailable to people that ingested it. So now we can also put these other puzzles into place. When I was first beginning to study lead and coronary heart disease, I came across this on the website of the National Heart, Lung, and Blood Institute. The gain in longevity has been remarkable. Between 1970s and 2005, the life expectancy of the average American increased by 6.6 years, 4.7 years. Over 70% of the increase is due to reductions in death from cardiovascular disease. We can attribute this remarkable improvement in large part to NIH research. Well, I contacted somebody at NIH and asked for a citation, and they apologized that they didn't have one, that this had been put up by a previous director, and they took it down. I think the real surprise here is that it's likely that NIH-funded research had relatively little to do with this. Most of it was kind of an accident and unexpected. We can also look at the Framingham study, which was a landmark study to try to understand why we were so focused on these lifestyle habits, that is individual behaviors. Dr. William Cannell, who's one of the directors of the Framingham study, said the social and economic factors that might explain changes in an individual's diet, sedentariness, and smoking habits were important, but not the focus of Framingham. They were someone else's problem. That is, they weren't interested in the upstream factors or environmental factors that might lead to coronary heart disease. They were only interested in what happened in the clinic setting. And this really, I think, represents one of the most troubling parts of our health system in the world, but let's say in the United States for a moment. And that is the vast majority of our resources, the vast majority of our research investments goes to what happens in the clinic setting after people are already sick. Less than 5% of NIH dollars go towards prevention. Less than 5% of our health dollars go towards prevention and public health. And yet, what kills us today are largely man-made diseases that is potentially preventable. Now, if we look at from a different perspective, what led to the decrease in deaths from coronary heart disease? You can see here that there were estimates for treatment in dark blue and risk factors like reduction in smoking, reduction in hypertension, and then in white, unexplained. And you can see that the largest fraction were due to reductions in risk factors, that is prevention. So even though the vast majority of our health dollars and our research dollars goes towards looking at treatments, the majority of the benefit was reaped from declines in risk factors, that is prevention. In some cases, like in Finland, the greatest investments were made in prevention. That is, they invested more and they reaped more. 2/3 of their reduction were due to preventive risk factors. In the United States, in contrast, where relatively less investments were made in prevention, you can see less benefit from the reduction in risk factors. In fact, where most of our dollars goes is to treatment. Very expensive coronary artery bypass grafts initially, now stents. And David Jones, who's a medical historian at Harvard, has written a book about this and talks about how this emphasis in many cases was misguided by focusing so much on treatment, and that benefits were largely overstated. It's a great book to try to get your head around what's happened over the past 3 to 4 decades, and to try to understand why 45, 50 years ago, we had leaders at NIH and elsewhere talking about how we can begin to control or even eliminate coronary heart disease, but the vast majority of our effort and resources and investment over the past 3 to 4 decades have really been about treating coronary heart disease, very little focus on prevention. And what focus there has been was largely about slapping people on the back of the hand and telling them to get their behaviors straight. We can also look at this more generally. This was a study out of the Netherlands where they found that what was the impact of high risk or clinical approaches and low risk or population approaches to the overall declines in death, not just for coronary heart disease, but more generally. And approximately what Johann Machenbach found is that approximately 3/4 of the health gains were achieved by population strategies and approximately 1/4 by a high risk or clinical approach. And yet, as I mentioned, the vast majority of our research dollars and our health dollars go towards the clinical or high risk approach. So Jeffrey Rose asked the question, why is so much of our research devoted to the mechanics of dying and so little to the scientific, social, and economic basis of prevention? And that's the question I'd like to leave all of you with today, because it's clear to me that until the community rises up and demands it, we're not going to change the way we address disease in this country or even around the world. We'll continue to focus on treatment because that's where you can privatize or monetize the benefits. And even though the social benefits of prevention are tremendous, it's hard to monetize them. And so when you're making decisions within a free market system, there really is no incentive to prevent disease. Thank you.
Thanks so much, Bruce. There are a few comments and a question. So starting with the question. So this is from Matt Shasher. Given your analysis of the data on increase in CHD from even low levels of lead, I would be interested in your thoughts as to public policy implications, which I think you've been trying to address at the end of this talk, but specifically much of the regulatory response until now has been focused on lead hazards in housing with children less than 6 years of age. Should we be rethinking this?
Absolutely. Now, some of this, of course, is going to be lead that was stored in people's bones. Probably the best estimate we can come up with is that, and this is from pregnant women, but it's the best estimate because Brian Golson did this novel study where he looked at Eastern European pregnant women who moved to Australia, and he could use the radioisotopes as a way to fingerprint where the lead came from. And so if we use that estimate, maybe a third of our the lead circulating in our blood is from past exposures. Now, that may be an underestimate in some cases. Women who undergo osteoporosis, for example, their blood lead levels increase by 30%. So it may actually be higher in some cases. So dealing with past exposures will be more challenging. But what we've seen, even as cardiovascular disease has declined, deaths have declined in more affluent, let's call them post-industrial countries, which isn't quite right. We've seen them increase dramatically in industrializing countries like China and India. So they're still on the upswing and focusing most of their efforts on the same lifestyle or behavioral choices and building cardiac intensive care treatment centers, as opposed to thinking more broadly about some of the exposures to air pollution and lead. So I think the policy implications are absolutely obvious in industrializing countries, but even, for example, in places in the United States, we still have a big problem with airborne lead. Most recently, Sami Zarin and his group did an analysis of the Reed Hillview Airport in Santa Clara County and showed that proximity to the airport was a risk factor for having a higher blood lead, as was living downwind. And like Maury and Aaron were talking about earlier, the particles from leaded aviation fuel emissions are much smaller than what you get in paint, for example. And that's true for other combustion engines as well. You'll see very small particles that in theory can go right up into the brain, directly into the brain. So there are still airborne sources of exposure in the United States that we've been neglecting for decades. Some hope, though, is that the Santa Clara County Council just in August had a meeting where Sami Zarin reviewed the report. There were 3 hours of community comments. Each comment was 30 seconds or less, 3 hours of that. Then by the end of that, the county council voted first to accelerate the closure of the airport, and second, to accelerate the ban on leaded aviation fuel in Santa Clara County. So after 30 years of almost nothing from the EPA, almost nothing from the FAA, we might begin to see some activities but it probably is going to start and has started at the local level.
Mhm. So coming back to if we talk about federal agencies and regulations and standards, we know that the knowledge about toxicity from lead exposure and lead in a person and the standards are basically two different continents, right? So we can have a standard set in the 1970s by the EPA and the data indicating that it's already 50 years out of date or 40 years out of date. And it just hasn't kept up. So waiting for federal agencies to correct this problem is, I think, a wishful thought. It's a very slow process.
I think that's exactly right. And I mean, I used to think naively that our jobs were to do the science and then go on and do another study, because of course there were policymakers out there, regulators out there who would pick up the baton and run with it, and wonderful things would happen. And I did it. My first study was trying to understand what levels of lead in house dust were dangerous for kids back in 1995. And finally, 25 years later, the EPA has finally been ordered by a court to use the science, not the costs or a cost benefit analysis, but to use the science to protect kids. That's 25 years. And that's true for most of the regulations. You have to have the science, no question about it. But until there's a crisis like the Flint crisis, or until the public is incensed about the failure to regulate, very little seems to happen.
So I have a couple of other questions, and we'll see if there are other questions coming into the Q&A. In the studies that looked at air lead exposure and criminal behavior, the two curves are superimposable for the rise and decline, like you were showing for gas and air in the same. But there was a lag of 18 years. And you also looked for the lag in your study. Right? You had an earlier blood lead level in adulthood, and then mortality over the subsequent 20 years or so, right? About that.
Yeah.
So in relationship to lead levels in blood, which I am going to talk about briefly in the next talk, in my talk, and the durability of the blood lead level as an assessment tool for history of lead exposure and lead accumulation in the target tissues. Again, we have kind of the equivalent of the federal discrepancy. We've got major differences between reside time of lead in blood and reside time of lead in the target tissues where the harm is being done, with bone being the major reservoir, as actually was raised by one of the people in the chat. So, Haynes also has earlier data, cohort data, well, back to the 70s in kids. So I wonder whether you would reanalyze your data using blood lead levels more proximal to a potential period of time of greater exposure and absorption and retention of lead. So, childhood lead versus mortality outcomes lagged over decades.
It's a great question. I don't know if you can get at that within Haynes, because it's not just the Haynes data. Some of it was some of the death index data were collected for parts of Haynes, but not others. So that adds a bit of a challenge. One of the other things, though, I wanted to just mention when you talk about blood lead, because I don't think we give it enough thought. We think of what happens in the blood is obviously important and is a good indicator, but it's an underestimate of exposure because it does not account for what is accumulated in the bone.
Exactly.
And I think that's a bit challenging for people to kind of wrap their heads around. So I tried to come up with an analogy I hope is useful. If we look at wealth or assets, and monthly salary as analogous to bone lead levels and blood lead concentrations. We can use a monthly salary to estimate people's overall wealth, and they're highly correlated, not surprisingly. But a monthly income may not give you a good indication of overall wealth or assets. And I think the same is true here for blood lead and bone lead. A blood lead concentration is correlated with bone lead. But if you look at a two-year-old blood lead level, a 2-year-old, let's say, who lives within half a mile of an airport, their blood lead levels will be higher than a 12-year-old who's lived in that same location for 12 years, right? So we're underestimating the impact.
Yeah.
And if you look at bone lead studies, somebody mentioned the veteran study, the veterans normative aging study in Boston that Howard Hu and others have been following for decades now. And yes, they've absolutely found very similar patterns, much more nuanced than what we found, not only an increase in blood pressure, coronary heart disease deaths, lowering of the threshold for arrhythmias. But what we would expect based upon that study and others is that if we actually had measures of bone lead from these individuals in our study, in the NHANES mortality follow-up study, we would estimate a greater contribution to the risk and a larger attributable fraction. So we think we're underestimating it.
Yeah. Well, thanks very much. I see Nancy is flashing me here that we're running late, and we're going to try and stick to schedule. So we're not going to have time for a break, but I'm going to launch into my talk. So let me share my screen. There. I think that should be visible to everybody. So both Aaron's talk and Bruce's talk really focused on adult consequences of earlier lead exposure and or concurrent, depending on which aspects of Bruce's talk we focus on. So it could be childhood exposure and brain effect 40 years later, 35 years later. Or it could be throughout life exposure and adult exposure and heart disease, cardiovascular disease. This talk is going to shift a bit. So I'm still in the adult group, but I'm also going to be talking about a diet because lead during pregnancy relates to not only the pregnant woman, but also to the developing fetus. So let me launch. This is the outline for what I'm going to say. I'm going to talk about lead sources, how it gets into the body, a mechanism of toxicity, and what happens during pregnancy, the epidemiology, what we can do about it during pregnancy, and what happens to the fetus. So the reason I'm going to start with the exposures is that everyone on this seminar knows about lead in paint, and we've heard about lead in gasoline and the residual exposures that we have currently because of our past use, especially in the 20th century. But lead is a promiscuous element. It likes to have partners, different partners, and we rarely encounter it by itself. It's always going to be a lead something compound, lead acetate, lead arsenate, lead carbonate. And every time it pairs up with a different anion, you end up with different characteristics. And being brilliant at creating things, humans find different uses for these different compounds. The easiest way to look at this small sampling of compounds on this slide is the color. So antimonate is yellow, arsenate is white, chromate, lead chromate is orange yellow, or it could be red, brown, gray. They're very poorly soluble in water at pH 7, and the stars next to the name of the compound indicates that there's more solubility. But most of them are easier to dissolve in acid. So what are the products that come out of them? There are hundreds, maybe thousands of products that have lead because it's used, again, not just for paint, or as tetraethyl lead, the gasoline additive, still in that aviation fuel. Stained glass as an insecticide, as an explosive, in batteries, etc. One more thing before I move on from these various lead compounds, which is when we talk about lead, we're exclusively only focusing on the cation, on the lead, and we ignore the anion. We ignore that the insecticide, the way it works, lead arsenate, may not just be from the lead, but from the arsenic, or that chromium, a part of chromate in a hexavalent state, is a carcinogen. Why do we just focus on the lead? Because most of the lead that we've encountered has been in the paint, and that paint, we think the anion was pretty benign, the carbonate or the oxide, but it's not always the case. And so in the adult world, where we're exposed to many different lead compounds, more so than in kids, it would probably be worthwhile for those in the field to be aware that the source may determine the toxicity profile. So to make all these different products, we have occupational exposures, and I found almost 40 different kinds of occupations you can be in that will result in exposure. But at least you get paid for that hazardous work. You may have hobbies, and again, we have about at least 20 hobbies that employ lead in whatever process is being used that can result in lead exposure. So being exposed, though, from work or from a hobby is really, I think, limited to a small part of the population. But the other products that we can come in contact with, not limited to those who produce these products. So I'd like to start with this particular example. These are long burning candles. They can burn for 12 to 24 hours. And to keep the wicks from flopping over and burning poorly, the wicks are enclosed in a metal sheath, usually zinc, but it could be lead. Even though the FDA banned the use of lead metal sheaths in these long burning candles in 2003, candles come in from abroad, or hobbyists might end up using lead because it's available and it's cheap. So what happens when you burn these candles is you cannot destroy the lead. It goes into the air. Thousands of micrograms of lead from a single candle can come into the air, or it can be inhaled, which we've heard a lot about, or it can land on the surfaces, contaminating those surfaces to levels above what is considered safe, at least for children and probably for adults. So from thousands of micrograms in a waxy substance to another waxy substance where there can be really tiny amounts of lead. A stick of lipstick might have 1 to 10 micrograms of lead in a whole stick. So you'd have to eat a lot of sticks of lipstick to get lead poisoned. And I think it would be safe to assume that you're not going to spread lipstick lead through kissing. But there are other products that have much more lead that are imported. We would think of these as cosmetics because they're used like eyeliners. They typically are black and made with carbon-based compounds, but they can also be made with lead compounds. And as a consequence, lead seems to be able to get into the body of the people who use it, resulting in lead poisoning. So again, these are forbidden in the United States. They're not supposed to be imported, but they do somehow arrive here. So getting away from candles and wax and cosmetics to foods that are contaminated, we go back again to tiny amounts of lead reported historically in apple juice, a couple of micrograms, maybe in a quart, to significant amounts of lead in tiny particles of these spices coming in from abroad, often from South Asia and Southeast Asia. Chili peppers, saffron are two of the most common, and they can have an enormous amount of lead. We don't know for sure how the lead gets into these spices, whether it's an incidental contaminant in the processing of the chili peppers, as an example, or whether it's an intended adulterant, because you can match the color with a lead compound. Lead is very heavy, lead is cheap, and the spice can be expensive, and so you've made yourself some money. candies from Central America, and the cooking utensils or the storage utensils or the plates that we eat off of, including very expensive china. This is a Haviland Limoges plate from a china set. And all of the paintings on here have lots of lead, which can leach into the food. And then there are products that are advertised as good for you. This one, made in the United States, is an herbal supplement that can have several hundred micrograms of lead per capsule, or more readily identifiable imported medicines, traditional medicines like Ayurvedic medicines from Asia, but not only from Asia, from other parts of the world as well. And then sometimes we have very sophisticated supplements that are imported. This happens to be imported into South Africa from India, but it's a very professionally made bottle with a child-proof cap, nice printing on it, nice shiny plastic, the ingredients on the back. But if I were to blow this up, these ingredients are illegible. And it had enough lead in each of these caps used to treat acne that put several teenagers in the hospital with lead encephalopathy. Okay, so these are many kinds of sources in many different domains, but it's not enough to be exposed to the lead. It has to get in to the body. We've talked about the inhalational pathway. We know about the ingestion pathway. Transcutaneous, getting through the skin, is very unlikely because only tetraethyl lead seems to penetrate skin significantly enough to raise the blood lead level, which means transmission to all the other organs. Of course, the exception to transcutaneous non-penetration would be a bullet if you're shot, and then direct passage, which is going to be relevant to pregnancy. So lead poisoning outside of occupational and hobby exposures is probably primarily through ingestion, at least these days. But eating lead is, again, just the beginning. Lead is not just coming in as an atom, it's coming in attached to things. And you have to separate it from what it's attached to, whether it's a paint chip, whether it's the spice. That's the process of digestion, and that requires the acid in your stomach. So the more time spent in the stomach, the more lead is going to leach off of it. And then it gets out of the stomach, goes into the intestine, where potentially it can be absorbed. But not all of it gets absorbed because it depends what else you ate at the same time. And if you're eating foods that contain essential metals like iron and calcium, that competes with lead for the pathways from the intestinal lumen, the center of the intestine, through the wall of the intestine to get to the blood and go from there. So what we have is you can eat lead once, but only a little bit will be dissolved. You're not likely to get lead poisoned from that. And even if you eat it repeatedly, there can be competitors which will decrease the amount of lead that you do absorb after digestion. So typically you have to eat lead repeatedly in order to get more lead into your body than you can excrete. So how much lead is that? How much lead can you safely eat? Depends on what you're looking to try and accomplish. And typically it's to try and prevent lead from being in the blood at a measurable level above a certain threshold. 1971, a group of experts got together and said that was 300 micrograms of lead that a child could eat in order to not get a lead level above 40 micrograms per deciliter, which was the level where clinicians began to worry about the impact of lead on the child. In 2020, the FDA had a different level that it came up with, 3 micrograms per day for a child, 12.5 micrograms for an adult. And this was to try and prevent a child from reaching actually a level of 5 and an adult of level of 10. There's a big safety margin built into this number, typically a 10-fold safety margin. So the lead that you eat, that you digest, that makes it through all the competition and gets absorbed and gets in the blood, gets distributed everywhere in the body. And the distribution is different between adults and kids. So the vast majority of our lead, 90 to 95%, is in our bones. And in a kid, it's more 70%, which means more in the soft tissues, where we think the harm is primarily occurring. That's background. What happens to an adult who potentially can get pregnant or is pregnant? So I think of the effects of lead. So lead toxicity, as opposed to a measurement of lead in a blood sample, let's say, or as an exposure, can be thought about at three different levels. The first and fundamental is biochemical, and then subclinical and clinical. Subclinical means an organ is affected, it's not working the way it should, but a person doesn't know. Clinical means they've got symptoms, they've got a headache, a stomach ache, they can't concentrate. This is the only biochemical slide in this talk. So on the top here in the red, this is a blood vessel. There's blood in it, and there's calcium, which belongs in it, floating here, but there's also lead. On the bottom in the white is the inside of the cell. The white is the cytoplasm, and all the way on the bottom is the nucleus. So calcium gets into the cell from the blood through a channel, a tunnel that the cell has produced that pokes through the wall, the cell membrane. And it's just supposed to fit calcium. But it turns out lead, which is much heavier as an element, its mass is 5 times as much as calcium. But the lead is packed so tightly in a little ball that its radius, if you think of it as a circle, the radius is smaller than calcium. So it can squeeze through the calcium channel and get into the cytoplasm. And here's one example of its effect there. Aaron had talked about the effect in the brain and neurotransmitter release and actually receptor placement with the lead competing with essential trace elements there. This particular example is occurring in every cell of the body. Calmodulin is like a master regulator, one of the master regulators inside the cell. It binds calcium, like the cal part of calmodulin. And when calcium binds, it turns on the activity of multiple other proteins. And cascading events happen in both the cytoplasm and in the nucleus. So a critical protein. When lead successfully competes with calcium and binds to the calmodulin, it diminishes the calmodulin activity. It changes it detrimentally for the cell. There are hundreds of proteins like calmodulin that have function, that are essential, that when lead binds to it, that function diminishes. So that's the biochemistry of lead. It can get into a cell. One of these mechanisms is through the calcium channel, but there are other ways it gets in the cell. And then it binds to places where it doesn't belong, and it causes more than mischief. So when we move up from the laboratory to people, to organs, and look at dysfunction, we needed a measure to assess the risk. And we chose lead in blood because it's convenient to get lead from blood to measure, and there's minimal harm involved in that needle stick. And because we've done it for such a long time, we have so many studies, we have a good sense of the correlation between blood lead levels in groups of people and various health outcomes, detrimental health outcomes. But if I were to take a group of lead atoms and inject it into me, lead atoms that I could follow, because I labeled them somehow, I would see that half of those lead atoms are gone from my blood in 3 weeks. Where did they go? Well, some of them went out through the kidney and were excreted in the urine. A few went out in the stool. Maybe a few were incorporated into nails and hair that were forming. And the rest that stayed in the body went everywhere else. They went to all the soft tissues, and they went to bone. If we were to look at the lead atoms that made it into the brain that I injected, half would still be in my brain 1 to 2 years later. And I'm extrapolating from studies not in humans, where we didn't do serial brain biopsies. And this is from animal studies. And when I look at how much of those lead atoms, how many of them are still in my bone, half are still there years to decades later. We're only measuring this compartment. And Bruce was talking about this in the Q&A. Whereas we really want to know this major reservoir, that 90 to 95% of adult lead would be found here, and to see what it correlates with in terms of health outcomes, because it's continuously dripping lead out of the bone, back into the blood, off to the soft tissues without necessarily having an impact on the blood lead level, unless in certain circumstances, there's an accelerated release of these bone lead stores back into blood, and we can detect that accelerated amounts. And of course, the lead atoms, they're really indestructible. We don't know how long a lead atom survives before it turns into gold. It's somewhere between 10 to the 38th and 10 to the 189th years. 10 to the 38th is 10 with 38 zeros after it, years. So we do use blood lead, and I'm using the blood lead here in a categorical sense. You know, I've written or drawn these hard lines between different blood lead levels and different effects, but they're really not hard. They vary across populations in terms of the sensitivity of individuals to lead at various blood lead levels in their blood. And there are many reasons for that, including the reside time of lead in blood. But they're handy to generalize. They've also changed over time, because the more information we had about lead effects, the lower and lower these lead levels have been labeled as being related to dysfunction of various organs. So IQ at very low levels, blood pressure starting to rise with elevated blood leads. And I have these question marks because I think these are, again, examples of the flux in the interpretation of blood lead levels in groups of people. The more we know about lead, the lesser the amount it takes to be hurt by it. So this is subclinical. This is clinical. The person knows something is wrong. They can't think straight. They're depressed, they're anxious, they have pain in their muscles, their joints, intestinal pain, constipation, peripheral weakness, their arms and their legs are getting weaker. And that progresses to the brain, also getting weaker to the point of coma, encephalopathy, and the potential for dying from it at very high lead levels, which in this country we see rarely these days. And at least in kids, we haven't had a death attributable to a blood lead level of any kind for more than a dozen years. That's not true in the rest of the world. And apparently it may not be true in the adult world. So what about lead in pregnancy? So first of all, it seems to affect the ability to get pregnant. So I've only cited two of multiple studies that are out there, but in this particular study from Finland, a lead level of 30 or more was associated with a delay in being able to get pregnant in people who intended to get pregnant. This study out of Mexico found that 10 micrograms per deciliter was already a dividing line, that you were 5 times less likely to get pregnant if you had a lead level over 10 compared to under 10 if you were trying for a year. If you did get pregnant, what happens? Well, one of the things to look at is blood pressure. So we know that in kids and in adults, a rising lead level is associated with rising BP. And Bruce also mentioned this. And we see the same thing happening during pregnancy, but potentially at a different lead level. And this is not clear to me exactly how different, but at least in this one study that I'm citing here, hypertension, meaning 140 over 90 or greater, is defined by the American College of Obstetrics and Gynecology. It didn't take much lead as measured in blood to be associated with hypertension. So not just a rise in BP, but a level of concern that requires more marked interventions. So hypertension by itself has bad outcome potential, as was the substance of Bruce's talk. But hypertension in pregnancy can combine with other organ dysfunction to be very serious and the combination is called preeclampsia. So typically it's hypertension, so high blood pressure, in association first usually with the kidney, and we can see the manifestation of that by the appearance of protein in the urine of the pregnant woman. And it's not a good sign, and it increases the risk of mortality, both for the pregnant woman and the demise of the fetus. And it requires immediate intervention, often requiring the delivery of the fetus if the gestational age of the fetus is long enough to give the possibility of survival ex utero. So is there an association between preeclampsia and blood lead level? With the exception of one study, the other 10 studies that have looked at this, that were included in this meta-analysis, said yes, lead is a factor for the development of preeclampsia. Preeclampsia meaning hypertension plus other organ disease in the second half of pregnancy. So another organ, back to the brain, has also been looked at in a few studies. So blood lead levels in pregnancy and psychiatric disorders. This is looking again in the second half of pregnancy primarily. First, just in women of childbearing age, that the higher the blood lead, the higher the risk of having depression or panic attacks. During pregnancy, also looked at specifically, one study from China in 2016, where the mean blood lead level of the cohort that was examined was just about 4 micrograms per deciliter. And an interesting thing was observed here. So as in other studies, a rising blood lead is associated with a rising dysfunction. So here we have a depression scale, and here we have an anxiety scale. And here we have blood lead levels going between 0, even though it says 0.4 here, that's a transformed number. In arithmetic numbers, it would be a lead of 2.57. So the higher the lead level between 0 and 2.57, the worse the depression, the worse the anxiety scores. But above that blood lead level, it was a flat line. So there was no further impact in this study from China on the development of depression and anxiety at these levels of blood lead. A second study from Japan, more recent, looked at a very large number of women. And here they're using a broader psychological distress scale. And they compared the blood lead levels in women who scored high on the scale, meaning more distress, versus those that had very low scores of distress. And they found no difference in the mean blood lead levels. But the mean for the whole group was only about 0.5 microgram per deciliter. So maybe there is a threshold for blood lead effects on depression and maybe for other psychiatric problems as well. Or maybe it's an issue of the scales. So the depression scale, is it sensitive enough to pick up effects of lead at very low levels of blood lead? All of this is open to future research. So we move on. If lead is harmful during pregnancy, who has it? So here comes a series of numbers. From the World Health Organization, based on 2017 census estimates in the US, 76 million women are of childbearing age. From the Guttmacher Institute, during around the same era, 5.5 million births. And from the CDC and Haynes data, 0.7% of women of childbearing age had a level of 5 micrograms per deciliter or higher, and the CDC considers that to be the reference level for intervention. So if we multiply the 0.7 to 76 million or to 5.5 million, we find the following things: that we can estimate that about 500,000 women of childbearing age had a lead of 5 or more. And if we only focus then on those that gave birth, we can estimate that about 40,000 newborns have the potential to have a lead of 5 micrograms per deciliter or higher. How do I know newborns when I'm only assessing lead during pregnancy? And the answer is from studies like this. Here, women at the time of delivery had their blood lead level measured, and the baby's blood lead, typically in cord blood, was assessed at the same time. And we see pretty much a straight line relationship. The higher the blood lead in the mother's blood, the higher the blood lead in the baby's blood. So for example, if I had a mother who had a blood lead of 30 at delivery, and I went up to find her circle in this cloud over here, and I went across to the babies, it would also be very close to 30. So maternal blood lead during pregnancy is a predictor of fetal blood lead, especially in the last trimester for sure, and earlier on we can surmise. So given these numbers, how many newborns were actually identified as having elevated lead at birth using the standard of 5 micrograms per deciliter? And as someone noted in the chat, that number has dropped for kids from 5 to 3.5 micrograms per deciliter as of 2 weeks ago. So here's what the CDC and the ACOG, the American College of Obstetricians and Gynecologists, put in their guidelines for prenatal lead screening. Prenatal health care providers have to give anticipatory guidance. That means teaching about sources of lead and how to avoid it. They should assess at the first visit whether a woman is being exposed to lead. And that's a questionnaire. And if the answer to any of the questions on the questionnaire is yes, then to get a blood sample for blood level measurement. And then if it's 5 or more, intervention. Guidelines, 2010 from the CDC, 2019 from the ACOG. So are these screening guidelines for finding lead-poisoned pregnant women being followed? So I looked through the literature to see how many studies had assessed whether guidelines were followed for this purpose. And the answer is I found one. And this is the one. It's a 2007 audit that came out of the New York State Comptroller's Office, where out of 2,100 plus OB providers in their registry, 100% questionnaires, of which they got 38 back, 9 saying, why did you send this to us? Your registry is out of date. And of the 29 remaining respondents, 12 said, we do not assess for risk. So 70% of those who received a questionnaire screened for lead during pregnancy. Screened, that's just a questionnaire. And presumably a smaller amount would have used a blood lead as the screening method. So if screening was done and a blood lead was obtained and it was greater than 5 micrograms per deciliter, what's the treatment? And the treatment follows the same 4 steps that we use for anybody else. Identify and eliminate the source of exposure if it's external. Modify the behavior that brings it into the person. So we can't stop breathing, but if you're in an occupation with exposure, you should be removed from a site where the air levels of lead are too high for that person to inhale, even potentially with safety gear on. And we were talking about the masks and COVID masks. So there is respirator gear that's required for people who are working in air contaminated with lead. So behavior modification, typically that means not buying things that potentially could have lead, not eating those things, and not putting things in your mouth. Maximize the competitors with lead. That's the calcium, the iron, and their supporting vitamins, vitamin D for calcium and vitamin C for iron absorption. And the last step is chelation. So we always talk about nutrition in childhood lead poisoning. Does it really help during pregnancy? I think there's one really definitive study and it looked at calcium in particular. So in this study, women received 1,200 milligrams either of a calcium supplement, a calcium carbonate, I believe, or they received a placebo, starting at the, basically in the first trimester, at the beginning of the pregnancy. And the blood leads were examined. And we see that the blood lead levels in the group that was treated with the calcium dropped more and stayed lower than in the placebo-treated group. So lead levels decline typically in most studies in pregnancy in the mid-trimester, and then go up towards the end of the pregnancy again. And calcium was able to mitigate the shape of that curve, making a steeper fall and not as much of an increase in the third trimester. We have no studies comparable to this for iron supplementation. The last part of treatment was chelation therapy, and we have extremely limited data, and I'm going to go through this fairly quickly. So chelation just means giving a chemical that will bind the lead and hopefully result in its excretion. This is a chemical model of one of our drugs, calcium disodium EDTA, that binds the lead in this kind of ring structure and is excreted in the urine. This is what happens. This happens to be a study of kids who had high blood lead levels, received calcium EDTA for 5 days. Their blood lead levels plummet. And then when they finish treatment, they rebound, usually because not new ingestion is occurring, but bone lead is being released, replenishing the lead in the blood. Not as high as it was pre-treatment. And then slowly, 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 the blood lead declines without further ingestion. In pregnancy, we have no controlled trials. In the literature, there are fewer than 24 cases for 40 years. My own personal experience is much greater, and it's overwhelmingly with calcium disodium EDTA, and it appears to be very safe, at least the way I use it, without toxicity to either the pregnant woman or to the fetus. And if we lower the blood lead level in the pregnant woman, we successfully lower the blood lead level in the fetus as determined at birth. So the 4 steps for dealing with blood lead levels during pregnancy is, if it's under 5, it's anticipatory guidance, right? That's even before a blood lead level is measured as appropriate. And if it's over 5, eliminate the source, change the behavior, nutritional support, and the question of chelation therapy, which I think has to be done in conjunction with someone who's had experience with it. I think I have a few more minutes, and I'm going to continue to talk about the impact on the fetus. So the first question is the fetus exposed in this closed bag inside of a person? And if it is exposed, is it hurt? And how do we treat it? So there's no question that lead accumulates in the fetus. This is a cadaver study of fetuses done more than 50 years ago, showing that as pregnancies progress, the amount of lead in the fetus increases asymptotically. It's curvilinear. And this curve, the shape of the curve, is identical to what's happening with calcium accumulation in the fetus. So the answer to the first question, can the fetus be exposed, is yes. The placenta is not a barrier to lead. It crosses freely. And as pointed out in a few slides back, maternal and fetal blood lead levels are very close to the same. Second question, is it harmed, or is somehow the fetus who's developing more resistant? So we can look at all of these different domains. We can look at, does it survive? Is it born on time or early? If born and on time or even early, are the organs complete? Is the size appropriate? And even if the baby is born alive, looking good, are there more subtle impacts from the lead exposure in utero, as in on cognition? So for fetal loss, this is a study from 1911, where a proto-epidemiologist collected data on housewives, mill workers, women who had worked in a lead-based industry before they got married, and women who continued to have lead exposure through work. when they were married and became pregnant. And this is the fetal loss rate. So it triples as you get to women who are having ongoing lead exposure during pregnancy. We don't know what their blood lead levels were. We only know that more exposure was associated with more fetal loss. And it was well known, at least in the 19th century, if not earlier, that if you took a concoction containing lead, you could induce an abortion, but potentially you could also induce your death. Much more modern, and I think, again, definitive study, this is out of Mexico, looked at what happens at higher lead levels in women in terms of spontaneous abortion, so losing a fetus in the first half of pregnancy. And they found that if you compared women who had lead levels between 0 and 5 as your base group to those that had higher levels, the chances of having an abortion doubled again, doubled again. But what happened under 5? You know, we're always now worried about what's going on at less than 5. And there is one study that looked at that and found that the mean blood lead levels in the women who lost a fetus between weeks 12 and 20 was indistinguishable statistically from the women who had fetuses that survived the first half of pregnancy. So blood leads under 5, not clear that it has an impact on increasing rate of spontaneous abortion as you go from 0 to 5. But that wasn't exactly the way these researchers designed their study to answer exactly that question. What about on the anthropomorphic side of it? Yes, babies might be a little bit earlier in their delivery. They might be slightly smaller in height and weight and head length and weight and head circumference, but not massively so. Not small for gestational age babies. And in terms of major congenital anomalies, not associated with blood lead levels under 40. What about cognition? Bruce Montefiore. This is a composite analysis of 7 longitudinal studies that showed that higher blood lead levels in this group of 1,300 plus kids lower IQ scores with no threshold, apparently, as soon as you get above 0. What about in utero? Well, we have a similar study that actually looked at plasma lead with the plasma lead levels in the pregnant women's blood being measured in the first trimester. And what we have here, plasma lead on the x-axis, and for the babies that survived to age 2 years of age, had their baby IQs tested. And we see the same basic shape of the curve. First trimester exposure to lead and a decrement in the cognitive scores of babies at age 2, infants at age 2, toddlers at age 2. I get the right word. So throughout the pregnancy, the brain seems susceptible to the effects of lead. What do we do for the neonate? Well, we stop the exposure as soon as delivery occurred. And for women who have blood lead levels of 40 or more, we would discourage breastfeeding until the lead level is below that. And potentially babies that drinking the milk of mothers who have leads in the 20 to 40 range also may be drinking significant amounts of lead that will be observable in their blood lead concentrations. We want to make sure that they have the right nutrients enrichment. That's trying to mitigate the effects on the brain. I don't know of any studies that have looked at that. And finally, what about chelation? Can we give our drugs to a newborn? And the answer is, again, we have very limited data, but probably we can. And it's been done. But here's another modality. Take out the blood of the baby and put in new blood, exchange transfusion, which to me seems a very aggressive method, but is also effective at lowering the blood lead level substantially. Does it remove the lead from the rest of the baby? Hard to tell. And the same thing is true for a course of chelation. In general, a single round of chelation therapy in anybody gets out a small percent of the lead that's in that person due to chronic exposure. So summary, last thing, Lead is harmful to all living organisms. It's not just people. Every organism that's been looked at is exclusively hurt by lead and not helped by lead. Pregnancy is not a protected time. It's a time of increased risk, even to a potentially lethal outcome, not directly from the lead, but lead is a contributor to the development of preeclampsia and eclampsia, and that the fetus is not immune. I don't know if we have any questions left and time for questions. Nancy, what do you say?
I think we could certainly take one or two. We are just about out of time. I see there is a question or two in the Q&A.
Hello, Dr. Markowitz. There are two questions in the Q&A. The first one is, can you hear me?
I can.
Okay, you can't see me.
I cannot. Okay.
I
There you
think there
go.
we go. Figure it out. The first question is, we currently have no program from Jane. We currently have no program to cover the environmental costs and investigating lead source for pregnant women. Do you have any suggestions on funding?
Yeah, so this is a political issue, right? Where does the money come from? It comes from the government. Where does the government's money come from? It comes from taxes and fees. So it's a chain that has to be worked out, and that requires advocacy. And the advocacy, first and foremost, is going to come from departments of health at the county level, because they're the ones who get the command from the state level, do this, do that, without necessarily having the funds coming along with the commands. So that has to be pushback from the levels for the people who are working on the ground, dealing with the issue person by person within the communities. But it can be helped by all other people who become aware of the issue of environmental health and environmental diseases, back to the public health problems and how the funding has to improve in that domain for the entire population to benefit. So not just during pregnancy, but throughout. So the question is really much broader than the issue of dealing with the exposure in pregnant women and going out to the houses to do the same kind of investigation that is currently done for kids that are young.
The next question that we have is regarding bone lead. Can lead be released from the bones during childhood? I know it can be released during pregnancy, breastfeeding, and elderly. Thank you. And this question is anonymous.
Yeah, so lead is two ways, in and out of bone. The problem is for the lead that it gets into bone that is incorporated in the crystalline structure that makes your bone strong, the hydroxyapatite. That's where it stays for a very long time. So anything that causes the bits of bone to dissolve, which is happening continuously in us, right? You know, we don't have dead bone. I'm talking about adults. Bone is alive and it's always turning over. New bone is being made, old bone is being removed. With that old bone being bone that's been traumatized just from the activities of daily living. They're like, think of it as tiny little breaks, little fractures, but so tiny that you're not aware of it. So when that old bone is being replaced with new bone, whatever was there can come into the circulation, and that is how lead comes out of the bone, back into the blood. And that can be accelerated during pregnancy if the pregnant woman is not having enough calcium coming in. Fetal calcium demand, remember, it goes with a curve like that. It increases dramatically, especially in the last third of the pregnancy. That calcium is, like the lead, is coming from maternal blood. So either her calcium is coming from the food that she eats, or it's going to come from her bone. So if she's not eating enough calcium, then she's going to meet the demand of the fetus from her bones. And when you dissolve it for the calcium, whatever's there comes out, including the lead. So the lead levels can go up for that reason. There are diseases that also cause an increase in bone resorption, which is our term for those little dissolvements of bone. And that can accelerate the release of lead to the blood and increase the blood lead. And we've observed that in people, not just with diseases like hyperthyroidism, hyperparathyroidism, but also trauma, severe trauma that results in your being immobilized, paralyzed, either temporarily or permanently. That's a signal to the body to, you don't need all this calcium and phosphate in your bones. So you start releasing it. And if there was lead there, that's also released. And basically that's occurring in a micro way during osteoporosis, the progression of osteoporosis.
Thank you for that answer, Dr. Markowitz. Thank you for your entire presentation. I'm afraid we do have to stop there, but our heartfelt thanks to all three of you for 3 outstanding presentations. And I want to thank again the New York State Department of Health and our own staff, all of whom together made this program possible. But most of all, we all want to thank you, our attendees, for joining this first ever National Lead Conference. And remember, if you want to watch again or share with a colleague, this session has been recorded and you will receive a link as soon as it's available. We do ask one favor though. Please, there is a post-event evaluation we'd like you to fill out before you leave the meeting. Your comments obviously will help us in planning future programs. And finally, I would just like to share with you a quote from Theodore Roosevelt. Far and away, the best prize that life has to offer is the chance to work hard at work that is worth doing. So in closing, I know as healthcare professionals, you believe as we do that this work is more than worth doing. In fact, it's essential. So carry on, stay safe, and thanks again for joining us. Goodbye for now.
Thank you, Nancy and Maury.
And thank
And-
you,
and
Bruce and
others.
Aaron. Yeah.
That was terrific. Well done, everyone.